Okay, so here we have our normal and our mutant protein strands. I filled in some of these already, the mRNA, with what we know based off of the codons. So methionine, our start codon can only have AUG as the mRNA, and then UGG for the next codon. Our third codon in the normal strand, it starts with UU, but we don't know what the third letter is. It can either be a U or a C, so we have two options that can fit here. Then an A and a C can fit here, any letter, and then an A and an A go here. We don't know what's here, GG, and we don't know what goes here. So for the mutant strand, we can do the same thing. We have AUG, UGG, AU, but then we don't know what this is. A C and A, we don't know this letter, and then our stop codon, all stop codons start with U, but we don't know what the other two letters are. So since we know that this is a frame shift mutation, we can try to figure out, well, is this an insertion or a deletion? So we'll look and compare what mRNA we have so far. AUG, UGG, and then up top we have a U and an A. So if we look at this, we can tell that it is not a deletion because there's no A in this spot, it's a U. So we can assume that this A was inserted, and so that means that this U is supposed to be where that U is. So now we can determine that this is supposed to be a U, and that fits right there for isoleucine. C, which fits there for phenylalanine, and then A and C, so we know C goes here. That means U goes up top. And these last two letters in our stop codon are a U, which works perfectly. A fits there, and an A fits there. But we don't know what these two letters are because our mutant protein stops. So we can just assume maybe this will be an A, and maybe this will also be an A because they both fit there. So now we have our normal and mutant mRNA. Basically what an insertion does is it shifts all of the remaining mRNA to the right. So if we were to take this out, we would have the same letter sequence as top, but when we add this in, it shifts all of it, so therefore it changes what the codon is, since it's read in pairs of 3. And that is how we end up with a non-functional mRNA down at the bottom.